So hello everyone. Thank you for having me today for this Innoprot webinar series. I'm really happy and honored to be here and share part of my PhD work. So today we're going to talk about how we can extract phycocyanin using spirulina mobilization in calcium alginate beads under a biorefinery perspective. But before we dive into the presentation, I'm gonna briefly introduce myself again. I will just add some points that was not mentioned before. So I'm a food engineer passionate about innovation and sustainability in the food system. And I'm also a proud mom of a sweet little girl who inspires me every day to contribute to a healthier and more sustainable future. Um, so I'm originally from Brazil, and I graduated with a food engineering from the Federal University of Technology in Brazil. And I also participated in the double diploma between this university and the Bragança Polytechnic University, where I later completed my master's in food quality and safety. And that's when I started working with alternative proteins, particularly microalgae, spirulina. So recently, I am in the final stage of my PhD in chemical and biological engineering. And I also had the opportunity to work as a research intern at the Plant Protein Innovation Center in the University of Minnesota in the US, where I could expand my knowledge on the extraction, characterization, and functionalization of plant-based proteins. So let me briefly introduce my research group as well. So we are an enthusiastic group led by Professor Filomena Barreiro. We are part of the Mountain Research Center and the Associate Laboratory for Sustainability and Technology in Mountain Regions. We are located in Bragança, Portugal, in the north of Portugal, a beautiful city of Bragança. And our group works on a wide of different topics, including bio-based polymers, natural and functional ingredients and applications, nano and microencapsulation, emulsion technology, and advanced fast prototyping techniques. These technologies are mainly applied to different sectors like textiles, cosmetics, leather, pharmaceutical, biomedical, food, and packaging. And my work is within the natural and functional ingredients and applications and emulsion technology area. So let's start by looking at the bigger picture and understanding why this research matters. So according to the United Nations, the world population will achieve 9.9 billion by 2054, and around 68% of people will be living in urban areas by 2050. So we have a growing population. So with the growing population, we also have a growing demand for food, especially for food quality protein. And many of this protein that we use currently, like soy, eggs, and dairy, they are also among the major food allergens. So we need to find new alternatives to feed people. Also, we need to preserve our land, water, and natural resources due to climate change, making it clear that the way that we are producing our food today, we need to become it more sustainable in the future. Consumers are also evolving in their preferences. So we are seeing an increase in vegans, vegetarians, and flexitarians, driven by concerns related to health, animal fare, and environmental impact. Also, we see the trend of consumers becoming more aware of healthy habits. So now they are demanding healthy and functional ingredients. So taken together, these factors, we see that we have an urgent need for developing innovative ingredients that are not only nutritional, safe, healthy, and functional, but also sustainable, so that we can meet the food future demand without compromising the environment. And this is where microalgae, specifically spirulina, become very interesting. Spirulina, which is actually a cyanobacterium with the scientific name of Limnospira platensis, is one of the most cultivated in the world. And according to the Food and Agriculture Organization, the global annual production is 30 tons of biomass. It's been used as food for centuries, and it's used worldwide as food and feed. You can find spirulina in the market as powder or tablet and capsules. But why spirulina? Why did we choose spirulina? Why spirulina is so important? First, spirulina is highly effective at carbon capture. Approximately 2 tons of CO2 are consumed during the production of biomass. 2 tons of CO2 produces 1 ton of biomass, while 2 tons of oxygen is released as byproduct. And second, spirulina does not need arable land to be cultivated, which means that it does not compete with food chains. From a nutritional perspective, spirulina is considered a high protein source with protein content ranging from 50 to 70%. And also it's a high quality protein source with a PDCAS of around 0.8. Not only protein, spirulina has a diversity of high value compounds like vitamins, minerals, and pigments, including chlorophylls, carotenoids, and phycobiliproteins, with which one of the phycobiliproteins, the phycocyanin, is the most valuable compound in spirulina. So spirulina is really interesting because since it has different compounds, it can be explored, it can be extracted in a biorefinery concept and a cascade extraction having different compounds, and also under a circular economy perspective, because some studies have been showing that you can use wastewater to produce, to cultivate this microalgae. So as I mentioned before, one of the most valuable compounds in spirulina is the C-phycocyanin, which sometimes I will call CPC. So phycocyanin is a water-soluble blue pigment protein complex and the most abundant in spirulina, representing around 20% of its dry weight. It's also relevant in the food sector as a natural food colorant alternative to the synthetic and conventional brilliant blue. And today we can find different extraction methods of this pigment in the literature, such as freezing thawing, which is the most used one, ultrasound, microwaves, high pressure homogenization. But these technologies are mostly energy intensive because in addition to the energy that they use in the process, they also need centrifugation steps at the end to separate the biomass from the extract. So they are also expensive and can lead to unpurified extract, and which will need further purification steps depending on the application and can increase the CPC costs. So during my master's, I was trying to encapsulate spirulina using a different several microencapsulations technology. As you can see here, I tried to use spray drying, and I also used the calcium alginate beads. During the encapsulation process, we noticed that the cross-linking solution was turning blue, and over time, the solution was turning even bluer. So then we analyzed the phycocyanin content, and we got interesting results. And since then, we have been studying the phycocyanin released from spirulina calcium alginate beads. So calcium alginate beads are systems commonly used to encapsulate, retain, protect, and release compounds. So this system can be used to release phycocyanin at food grade purities, facilitating the separation steps as no centrifugations will be needed at the end to separate the beads from the extract. But how the beads are formed? So the beads are formed by the diffusion of calcium ions into the alginate droplets. Gelation and cross-linking are mainly achieved by the exchange of sodium ions from the alginate with the calcium ions, forming the characteristic egg box structure. So this system has been extensively used for in protein delivery, showing relevant outcomes for retaining and releasing proteins from food products along with the gastrointestinal tract. And most of the studies addressing spirulina mobilization in calcium alginate beads do not aim at the phycocyanin release, but rather at increasing the spirulina or phycocyanin stability over time. So this work focused on the immobilization of the spirulina biomass in calcium alginate beads, focusing on the releasing of phycocyanin at food grade purities and analyzing the leftover beads to be further valorized under the biorefinery concept. So let's move on to the methodology. The beads were prepared from a mixture of spirulina biomass powder and sodium alginate at 2 and 4% at a ratio of 2 to 1. And they were formed by adding droplets of this mixture into a calcium chloride solution at the same concentration, 2 and 4%, which we're going to call PC2 and PC4. The natural pH of these extracts was in the optimal range of the phycocyanin extraction, which is between 6 and 7, as below 5 and above 8, CPC becomes unstable. 4 spirulina solvent ratios were studied, ranging from 1 to 33 to 1 to 125. After 24 hours of extraction at room temperature, the beads were separated using a stainless steel sieve, and the phycocyanin concentration was measured, the phycocyanin concentration and purity was measured according to these well-known equations. Allophycocyanin, which is another phycobility protein that can be co-extracted with phycocyanin, was also analyzed according to this equation, and their yield, both CPC and APC, was calculated based on their concentration in the volume of the calcium chloride solution and also the initial weight of spirulina biomass. We also calculated the efficiency of the extraction based on the yield obtained from this technique and the yield obtained from a serial extraction of freezing and thawing. We did this extraction until we achieved the maximum yield, and then we compared with the immobilization technique. So let's go to the results. This is how the process takes place. So the initial stage is the beads production. And after 5 to 10 minutes, the solution starts to turn blue, which means that the phycocyanin and also the allophycocyanin start to be released from the beads. And that occurs over time, a long time. So we suggest that since phycocyanin and alginate have similar charts at the pH we use for the extraction, they tend to electrostatically repel each other. So the phycocyanin, the protein pigment, tends to be released from the beads. The attractive alginate protein interactions are a lot weaker than the alginate calcium interactions. So the protein is released when the droplets of the alginate spirulina mixture are into the calcium solution. And at the final stage, after 24 hours of extraction, we have a dark blue solution, as you can see here. In this graph, we can see the phycocyanin yields and purities after 24 hours of extraction. So higher spirulina solvent ratios result in higher phycocyanin extractions yields, which is associated with the availability of the solvent to perform the diffusion of phycocyanin. So the higher the availability of the solvent is, the higher is the diffusion and consequently the extraction yield. On the other hand, higher spirulina solvent ratios can generate phycocyanin extracts with low purities, as the increase in the solvent ratios as the increase in the solvent ratios, just a minute, leads to extraction of impurities, yes. And this behavior can be observed in the obtained results as the highest, and as the lowest period were obtained in the highest spirulina solvent ratios. So higher purities were found when higher calcium alginate concentrations were applied. For all the spirulina solvent ratios studied, PC4 extracts presented purities higher than the established for food grade, which is 0.7, as you can see here. So the best conditions were those with the highest purities, PC2 at the 1 to 83 spirulina solvent ratio, and PC4 at 1 to 42 spirulina solvent ratios. So regarding the phycocyanin efficiency, we can see that higher efficiency were obtained at the higher spirulina solvent ratios, aligned with the phycocyanin extraction yields, and the best conditions obtained efficiencies higher than 50%. As we can see here, PC4 extract presented an efficiency of 55.1%, and PC2 extract presented an efficiency of 66%. Here in this graph, we have the analysis of the extraction over time. And as you can see, there is a significant increase both in phycocyanin purity and in phycocyanin extraction yield, considering 1 and 24 hours of extraction. Moreover, we can also see that the highest purity here was obtained at the highest calcium alginate concentration, PC4. And in general, the increase in the alginate concentration leads to a decrease in the diffusion rate of proteins from the gel. So the obtained results showed that higher alginate concentration impeded the diffusion of other proteins, such as the allophycocyanin, and enabled the phycocyanin release at higher purity ratios. We can see that here, let me show in this graph, that the allophycocyanin yield in PC4 is lower than in PC2 after 1 and 24 hours of extraction. So the purities obtained through this work were aligned with or even higher than the purities obtained from commonly used extraction methods in the literature, as you can see in this comparative graph. Here is our work, and we can see that we have higher values than ultrasound, similar and a little bit higher their values than freezing thawing methods, which are the most common used methods in literature. So regarding the spirulina calcium alginate beads, the leftover, what we have after the extraction, in general, their proximal composition, as you can see here, and the average size are quite similar, and relevant protein contents still remain in the beads, being higher than 25%. So they can still be valorized and to extract lipids or proteins, or even to be used as food supplement. So this work is already published. So if you wanna check it out and read the complete work, you just go online and search in Algal Research Journal and you can find it. And overall, the proposed technique enabled the phycocyanin release, as we can see along the process, at relevant yields and food grade purities. So it can be used as a food colorant, alternative to the synthetic one. And the beads can be also used as food supplement or for protein lipid extraction, as it has shown relevant protein content at the end. So although this procedure needs improvement, especially because of the extraction time, it's a simple and easy methodology which facilitates the separation of the extracts from the beads. So we don't need centrifugation steps, and it can reduce the phycocyanin costs, which of course should be confirmed by a techno-economic analysis. So I'd like to thank the head Innoprot SciTed for the opportunity to present my work in this webinar, and also the funding agency, the Foundation for Science Technology, the Portuguese Foundation for Science and Technology, for my research, my PhD research grant. The Aldikick company, it's a Portuguese company that kindly donated the spirulina biomass. My supervisors, Professor Filomena Barreiro from CIMO and Professor Madalena Dias from the University of Porto, and the host institutions. So thank you very much. I'm open to questions. And here in this QR code, you can also find this work, the link for this work, and other works that I publish in this field. Thank you.
Thank you, Samara, for this very interesting presentation for your work. We have time for some questions. If someone have a question, can open the mic and ask directly or write in the chat. Any question for Samara?
Okay, I started. Samara, thanks a lot for the presentation. It was really, really interesting. And okay, for us that we work with microalgae, I think is really interesting to know that there are more people working on that. So I have a question. Do you think that the result will be different with other strains of spirulina, or they will be the same?
No, I think it will be different. All other strains of spirulina, you
Yes,
mean?
yes.
No, I think it will be the same. I
Mhm.
understood it was a different species of microalgae. Spirulina will be the same because they mostly have similar cell walls. And here, when we dry the spirulina powder, when we dry the spirulina, we partially broke the cell wall. So we have this protein, they will be free to be released. And I believe that all spirulina strains will be the same, quite similar. But if we're talking about different microalgae, yes, it will be different. In the case, for instance, if chlorella is completely different because the cell wall of chlorella is made of cellulose. It's really rigid, so it's quite difficult. But in this case, it's impossible because we're not talking about phycocyanin, right? It's another kind of protein which is that is present in chlorella. But I believe that Gaudiata, it's another strain of cyanobacteria that also has phycocyanin, can also be used in this process.
You are going to try with other species or
Not for now.
not
Not
for
for
now?
now. Yeah.
Okay, thanks.
Thank you, Blanca. Any other question?
Well, I will ask it also as we are working with microalgae and I found your presentation super cool. I was wondering as if you were using commercial spirulina, but I understood at the end of the presentation that you are working with a company. So my question was related because I guess the concentration of phycocyanin will change depending on the growing conditions of the microalgae.
Mhm.
So is something that you have like evaluated or you are just focusing on the extraction process?
Yeah, no, we're just focused on the extraction process, and the spirulina is we got it from the Portuguese company. They produce there, and I think it's in lake. Yeah, it's in ponds. It's in pond. They produce in ponds, and they send us the sheet, and it's about 20%. It's about
Mm-hmm.
20% of spirulina dry weight, the amount of phycocyanin in there. But we didn't analyze it. Our focus was only on the extraction process.
Mm-hmm. Yeah, and it was interesting the thing that you were saying that other microalgae with a thicker cell wall will be different. So it is something that opens a lot of research around there. And another question that I have is like these beds will be incorporated directly into the food, or how is the concept of these beds?
No, the idea of this beads, yeah, it can be. I mean, it can be, but our main focus or was in the valorization of the beads as a protein. Like we can further extract. I did some trials of extract of using this bead to extract carotenoids and chlorophylls, and we can do that. So after having these beads, I can put in ethanol, for instance, to extract chlorophylls, and then I can have the same beads to and use different kinds of solvents to extract carotenoids and have a cascade extraction. But I didn't focus on that because we didn't have very good results. So I was only focus on the phycocyanin. But yes, we can use the same beads to pursue a cascade extraction and have different pigments, or even use these beads as to do the protein extraction afterwards or lipids extraction. So it's easier because we don't need to use centrifugation, right? So we get these beads, we put in the solvent, and then we just make a filtration, do a
Yeah.
filtration.
And you are talking a lot about that it can be applied to lipids. So have you tried, I don't know, lipids from microalgae or other sources?
No. I've never worked with the lipids. I just extracted to analyze the lipids content, but no, I've never worked with it.
Mhm. Okay, so thank you very much for the presentation. It was
Thank
very
you.
interesting.
Thank you. Thank you for the question.
Thank you, Pilar. Is there any other question? I have a question, Tamara. Regarding the working with the industry, you mentioned that you get the spirulina from a company.
Mhm.
Is that like a project with the industry on the or they're just giving in kind the materials that for your work?
No, they just gave us the material. It's from A for F group. I don't know. It's a very well-known group in Portugal, and they have this algae company, which is part of the A for F group, and they produce spirulina and other microalgae species. So they gave us a huge amount of microalgae. So we're really grateful to them.
Yeah, yeah, thank you. And also, I was thinking also the same as Pilar about the applications of the bits. Finally, is a product that we can have and how we can add it to functional foods. You have any idea how can this be added or used in a food matrix or something?
We've seen the bubble tea. Maybe it's an option for a bubble tea. We can add this in and increase functionality because phycocyanin has the color, but also the antioxidant properties. So we can maybe add this in beverages, or I would say the bubble tea is really cool. Maybe.
Okay. All right, thank you. Very, very interesting uh, presentation. Your work is very interesting.
Thank
Any
you.
any any other question? Well, if not, I think everything was super clear. And thank you very much for your presentation
Thank you.
and let us know everything. I don't know if Blanca, you have any comment or final remarks.
Mm,
No, just only to invite everyone for the next one next month. So we will send the email soon with the invitation for the next one that is it will be given by okay by the group from Peru. So
All
we will
right.
send you the invitation. Thanks,
Thank you very
Samara.
much.
thank you,
Have a
everyone.
great
Bye
day.
bye.
Bye.
Okay, have a nice day, all of you.
You too. Bye bye.
Bye.